somebody remind me to get this online before I leave. I got to download it on YouTube. I have a couple followers.
How many is it?
I don't know, like 8 or 4. All it is I never look at their comments because I'm like, all right. So, couple of things here. This is what I told my other little kids, and I want to make sure that you guys know. I am 90% positive. I haven't talked to the other bio team, but I am 90% positive that I'm 90% positive that you would be able to use your review or these notes for your final. 90%. So now we need Chromebooks down, people. We're going to get looking at this, okay? So if I have it highlighted, you need to highlight for me. So if I have it highlighted, I would highlight. Now, on that little Poggle thing, we saw a little bit of how the cell membrane was made, didn't we? Right? Okay. And today I'm going to talk a little bit more about that. But the cell membrane defines the cell's boundary. So it is, yeah, I mean, it obviously surrounds the cell. We are considered animal cells, okay? No animals have cell wall. So this truly is our boundary. So when I talk cell membrane, I will talk via animals, or via us is what we're talking, dealing with us, okay? So it's two main functions. It's the boundary. So what does that do? It keeps it together, separates it from the environment. So it's there to protect. Probably a bigger thing, maybe, we could say, is the fact that it is it keeps homeostasis, keeps the cell in homeostasis. So you've often heard, what's the control center of the cell? Nucleus. People would argue that that, in fact, is not true. The cell membrane is the control center. Because the cell membrane is the one that's keeping things, allowing some things in and out. It controls what comes in and out of the cell. It decides if it needs more. It can shut down permeability to keep harmful things out. So it decides some of it. Sometimes we'll decide what reactions will take place in the cell. The nucleus is more static. It's basically just holding the DNA. So, but people will call that because it decides what proteins to make. But just understand that if you're talking cell by itself, a lot of people will say the cell membrane is your control center. So it keeps things in what were you going to say, Kate?
Oh, nothing.
Okay. Keeps things in homeostasis. And what does that mean? How does it do that, I should say? It regulates what enters and what exits able to keep harmful things out. It'll shut down permeability if it needs to. All right. Oh, for Pete's sake. So, 1972, two scientists, two biologists by the name of Singer and Nicholson went ahead and proposed the fluid mosaic model. And I told little kids that's the year I was born. So it's 47 years ago. I'll do the math for you. Almost 48. So the fluid mosaic model is the best explanation that we have. Some things may have changed, but so far it's the best that we have. So there's 4 components that make up the cell membrane. These first 2 are the ones that I would expect if I ask you to draw and label the cell membrane, you would be putting these in. Carbohydrate, cholesterol, no. Do I think that you should know what they do? Yes. But so if I ask you, what are the main components of the cell membrane, you would tell me phospholipid and protein. Does that make sense? You don't have to tell me carbohydrate nor cholesterol. I will talk about all 4, and you'll need to know the jobs of them, but when I ask for the main components, the main one is phospholipid and protein. No, I'm not. I just thought it was nice, in passing, these are the people. I'm not going to ask you people names. These components are able to flow and change position. So it's able to keep that membrane intact, though. That's an important concept. So the mosaic is the components. And if I get a chance, I would like to see that little Amoeba Sister film that I didn't show last time. So yeah, keeps the membrane intact. Now it says that we need to draw. People, I'm going to draw it over here because I've got people that were absent, and the only way that they can see the drawing is if I draw it on this board. I've been drawing it all day on the other, so I hope I have enough room. And I hope it cooperates with me. I would draw 5. So I would highlight the fact that it forms a lipid bilayer or 2 layers. It forms a bilayer, which is 2 layers. And the first thing is you want to just draw how it is here. I would probably draw maybe 5 of these things. Ooh, hark. Who goes there?
We're going to steal your keys. We're going to go put your stuff in your car.
Oh. But one goes to, I'm podcasting. One goes to Monica.
This is all that's left. But
No, there's another container.
this is it.
No, there was another.
This is it. Total left. This is it. So you can just give this to her. That's
Can you
it.
put this in the refrigerator?
Yeah.
Are you sure from the other?
This is it. I got both crock pots.
This is it. Where's your keys?
We'll put them on.
I don't. Oh, the crock pots, but I don't want that cake. Nothing.
Okay.
If we can turn your podcast back on.
Oh. Okay. Sorry. Was it hard for me to get it through my head that that was it? Okay. People. She didn't like my hark. Who goes there?
You didn't look like you trusted her with your keys.
Well, I didn't know, like, well, okay, when she said I need your keys, and I thought, well, for what? I didn't want any of that cake or anything. But it's my crock pots. Oh, and my ladles. Remind me to yell out there if they've got my. I'm sorry, but this is the way it was, third block for the other ones. Hey guys, did you get my ladles? Yeah. Okay, and the spatula? The kids are laughing at me. All right. All right, people. So this is our phospholipid. Let's go ahead and we're going to say this area here is the phospholipid. Okay. Now, we're going to take this a step. Now, you people are going to have to draw in proteins at some. We're going to draw in proteins too. But we're going to take this a step further. I'm going to say this is inside the cell. I'm going to say this is outside. I want to know, what are we mostly made up of? Good. Water. Inside the cell, we have a lot of protein. Yeah. If I would have said biomolecule, and then now, let's look at this. I kind of gave it away. This is our phosphate head. People, I'm going to draw the proteins in right now. We're going to do that before I even talk about it. This is the phosphate head. This is our tail. Which ones are Which ones are hydrophobic? Yeah.
The tails.
The tails. Which ones are hydrophilic? The heads. Heads are always facing towards the water. Yep. Heads are always facing towards the water. This is a lipid, your fatty acid. Oil and water, does it mix? No. Are the lipids polar or non-polar? They're non-polar and the water is polar. They will always orient themselves like this because of water. End of story. So, in order, this is the school of thought I go with. In order for water, is water polar or charged? You just told me. Yeah. Do we need water in our cells? Yes. In order for that to happen, we have to have proteins. This is why proteins are so important. So we've got two different types of proteins, and we're going to talk about it. I'm going to draw it so I can erase it. But the little kids that were absent need this. Draw this. This is one of the proteins. This is what we would call, the type of protein this is what we call an integral protein. What do you notice about it? Is it going in just one part of the membrane or the whole kit and caboodle? The whole thing. So when I talk about water moving through, it's actually a special protein that's known as the aquaporin. And it's like a covert operation. Water's small enough, but it's charged. It comes on through here. These tails have no idea what's coming through it. So it's undercover. Covert. Covert operation. So these proteins are highly important. Also, on the surface are what we call peripheral proteins. Now let's see. There. Peripheral proteins. I think I spelled that correctly. I hope I did. Are they spanning the membrane, Carter? No. N-O spells no. And they are not spanning. They're on the outside of it, on the edges. So think of it. I have my vision. If I'm looking off to the sides, what's that called?
Yeah.
Peripheral vision. So it's like peripheral. It's on the,
Alpha.
yeah, it's on the edges. Yeah.
So is there like proteins in between every single one?
Probably. Not necessarily though. But there's a lot of protein in there. The other one I'm going to go is on the outside. Okay? If I can, oh, for Pete's sake, people. That's going to be a peripheral. But look what I've got. A little Y coming off of it. Yeah, like a little bean sprout. Now, that's a carb. So that's a special type of peripheral protein. I've got to go all the way up here, and it's known as the glycoprotein. You also have glycolipids. I'm not going to draw that. In between the phospholipids, you also have cholesterol. And I'm not going to draw the cholesterol. It's a hexagon. If you can draw it, great. I can't. But what that does is, we'll see what it is in a minute. It keeps its structure. So it helps to keep the cell membrane structure, and it acts as an antifreeze. So it keeps that cell membrane fluid as temperature changes. Keeps the cell membrane fluid. So some animals, if they are in colder climates, will have a lot more cholesterol in their cell membranes than others if they're in like warmer climates. Okay? So I'm going to erase this now. You do that, Mrs. Eckel. You do that. Okay. So this is what I was talking about. You've got two types of proteins. Okay, I got to get a move on. You've got the integral proteins, which are embedded in the whole membrane. It's like they're stuck in there. So they're embedded in the whole membrane. They're stuck. So they get stuck in the membrane. Or like they're embedded, whatever you want to say. Their main job is either they're a channel or a carrier protein, and they're just going to move substances across. Some of them can be enzymes, and I reminded you what an enzyme does. It speeds up a reaction. If I have it highlighted, it's going to be, it could be on your quiz on Tuesday. It will be on your test. The other type of protein is the peripheral protein. And this is what I was talking about. I think probably the better explanation would be that they are along the edges, but I didn't write that. I said along the inside or outside. I wanted you to know that they can be on the edges either side of that cell membrane, either facing inside the cell or outside of the cell. So that's why I wrote that. Understand that they're not stuck. A lot of them can detach themselves and move around. So these are the cell processes, and they make the cell do something. Maybe it's hormones, and they might open up the permeability and allow glucose to come through. They may shut down permeability. I mean, they could do a few things. They could do reactions. So it just kind of depends. But they make the cell do something. Carbohydrate, I need you to know that they are for cell recognition. Sometimes you might hear them as cell surface markers, but they are cell recognition. So every one of you has unique cell recognition. Every one of you has unique cell recognition, and we spent a lot of time last time talking about identical twins and that. But, so, if a foreign substance invades, it's going to shut down that, it's going to say, nope, not today, not on my watch. So then it shuts down permeability, and it doesn't allow anything to pass through. Cholesterol is our fourth one, and that's what I was talking about as far as the antifreeze. So this just regulates the fluidity of the membrane, and it bases it on changes of the cell's environment. And it gives the cell membrane some structure. And that's what, and you can kind of see it. So it's spanning here. Here's a carrier, in some type of integral, I don't know. So there's all these little proteins that you can see through here. Oh, integral, okay. Here's a carbohydrate, glycolipid. Here's a glycoprotein. Here's cholesterol in there. So how does the cell membrane stay intact? One word. What is it, people? Nope. No. How does it stay together? Say it. Good. Water. It stays together by water. That's what I was talking about. Stays together via water. And just like we said, inside and outside of the cell is mostly water. How does the water keep the membrane intact? Tails are hydrophobic. Heads are hydrophilic. So when I say hydrophobic, you would know hydro means what? Water. Phobic means
Fear.
fear. So these are water fearing. They don't like water. So they're always going to orient themselves towards the water or away from the water. The heads are hydrophilic. Hydro again means what?
Water.
Water, and philic means, that's Greek. That means, I heard it, I think, to love. So these are water loving. So they are facing towards the water. They don't mind the water, they like it. So whatever I've highlighted will be fair game for your quiz. So just realize that. So whatever I've highlighted is fair game for the quiz. So the next thing that we're going to do is we're going to set up our egg lab. So that's what has been smelling like vinegar.